Today, we will be discussing amyotrophic lateral sclerosis, also known as ALS. This presentation is by Johanna Kramer and Jake Nestor. Societal visibility of ALS has increased in the past 100 years, with famous Yankees baseball player Lou Gehrig suffering from the disease, renowned physicist and writer Stephen Hawking suffering from the disease, and the Ice Bucket Challenge, which took over social media in 2014 to raise money and awareness for the disease. ALS is a motor neuron disease that causes muscle weakness and atrophy throughout the body due to degeneration of both upper and lower motor neurons in the brain and spinal cord, respectively. There are a number of phenotypes that have their own speed of progression, as demonstrated in the figure to the right, including bulbar onset, which first is expressed in the lower portion of the face, and primary lateral sclerosis, which progresses evenly throughout the limbs. The disease eventually leads to respiratory failure and death, typically within 3 to 5 years from onset. This is due to disruption of the development of axons, which impairs transmission of impulses from the nerves to the muscles, leading to muscle weakness and atrophy. This slowing of transport of materials needed for the proper function of axons and motor neurons eventually causes the motor neurons to die. When it comes to inheritance, there are two types of the disease: sporadic and hereditary. Only 5 to 10% of cases are hereditary. In these cases, autosomal dominant mutations in a number of genes can lead to ALS. To date, 25 genes have been identified to associate with ALS. In particular, autosomal dominant defects in the SOD1, which is a super dioxide dismutase gene, are associated with approximately 20% of these familial ALS cases. Mutations in the C9orf72 gene are responsible for 30 to 40% of these familial cases. Because of the high number of complex gene interactions that can lead to ALS has no definitive cause. A total of 12,187 persons in the United States were identified with ALS. This comes down to about 3.9 cases per 100,000 people in the US. ALS is more common in individuals above the age of 50 years, although juvenile cases are much more common in hereditary patients. It's also found that men have a higher prevalence than females across all age groups and data sources for ALS, and that Europeans have a higher prevalence of ALS than people of other racial backgrounds. Relative gene products include mutations in several genes, including the C9orf72 and SOD1 genes previously mentioned, as well as a plethora of other genes. Up to 25 genes have been identified so far in association with ALS, but it's unclear how these mutations contribute directly to the death of motor neurons. Most motor neurons affected by ALS have a buildup of protein aggregates around them, but it's unclear if these aggregates occur as a cause of, or rather as a result of, the progression. Because it has proven difficult to pin down a single specific genetic culprit in causing ALS, we instead propose that we study the cause of one of the most common phenotypic aspects of the disease, i.e. the protein aggregates that form around the ailing neurons. We propose a study utilizing mice to test if the protein aggregates associated with ALS are directly or indirectly contributing to ALS, or if they are instead just a byproduct of the disease. Mice were selected due to their high degree of similarity to humans and relatively short amount of time needed to breed them. We propose an experiment in which we mutate mice in their SOD1 and C9orf72 homologous genes to induce ALS. Among the mice with ALS, we will cross them in order to receive a stock of mice with hereditary ALS. Among these mice, we will remove the protein aggregates that form around their affected neurons via surgery. Very, very small scalpels will be required. Among those mice that survive the treatment, we will continue to study them for continued symptoms of ALS and for regrowth of protein aggregates. If the protein aggregates do not regrow and the progression of ALS among the mice is slowed, we can conclude that the protein aggregates play a role in the causality of the disease. If the protein aggregates regrow or do not regrow, and the progression of ALS among the mice is unaffected, we can conclude that the protein aggregates are simply a byproduct of the disease, and further study will be required.